Host or hijacker. No one is safe. Look closely. A rosette of jeweled tentacles, each one tipped with a bead of light, like scattered dawn dew resting on a plant too delicate to hurt anything. Hold that thought. Because that dew is not water. It's glue. And this is the gentlest killer on this list. What you're looking at is 9 plants that evolved to break the one rule the living world takes for granted. That plants don't hunt. They all learned it in the same desperate place, bogs and sands so starved of nitrogen that sunlight was never enough, so they turned to meat. They lure. They drown. They count electrical pulses. One runs a mammal latrine, one fires in half a millisecond. We count up 1 to 9 from the most beautiful and passive to the most lethal and fast, and the deadliest is a killer too small for you to see. Host or hijacker, no one is safe. We start where beauty hides the most patient death. Number 1. The Sundew, Dressera. Those glistening droplets read as pure decoration, a plant wearing morning dew. Every shining bead is mucilage, a sticky, sugary glue secreted by the plant that attracts, ensnares, and later helps digest. An insect touches one and sticks, struggling only pastes it to more tentacles. Then the plant answers. Researchers have shown that struggling prey triggers electrical signals, action potentials that travel down the tentacle, comparable to, though not identical to, the impulses along an animal's nerve. Krausko and colleagues in 2017 demonstrated that these signals, together with the hormone jasminate gathering where real prey is caught, let the plant tell food from a raindrop. This is signaling, not thinking. The plant does not choose. Over minutes, the tentacles bend inward, driven not by muscle but by changes in the cell's internal water pressure, folding the victim deeper into the glue. And it is slow. Sundews do not snap. Only one Australian exception, Drosera glandulagera, catapults its tentacles in about 75 milliseconds. The rest bend over minutes, curling whole leaves over hours to days. The glue kills first. Mucilage clogs the tiny holes the insect breathes through, and prey often dies. Commonly within roughly 15 minutes, before the enzymes even begin the slow melt. But glue is patient, and patience only ranks number 1. The next killer hides the very same glue behind a soft, harmless shine. You would never guess the buttery little leaf is already dissolving whatever lands on it. Number 2. The Butterwort. Pinguicula, Latin for the little greasy one. A neat rosette of soft, pale green leaves with a buttery sheen, a single violet flower held high on a bare stalk like a shy wildflower. That shine is not dew. The leaf is carpeted in tiny stalked glands, each holding a bead of living glue. An insect lands, sticks, and glazes itself in adhesive as it struggles. A second gland type does the digesting, flat. Stalkless glands that flood the leaf with enzymes. Researchers studying the hybrid Pinguicula tina catalogued around 8 and found this butterwort ramps them up without the jasminate pathway most carnivorous plants rely on. In some temperate species, the leaf margins then creep slowly inward, over minutes to hours, but this is after the fact, not a snare. The glue does the catching, the plant is entirely passive. Researchers suggest the curling either pools the digestive fluid around the catch or helps shield it. The function is still debated. That violet flower stands high on its bare stalk, understood as a way to keep pollinators safely away from the killing leaves. And those leaves hide a stranger past. For generations across northern Scandinavia, fresh butterwort was used to curdle warm milk into a thick, tangy curd, a leaf that dissolves insects, turned to setting supper. A fungus gnat, a springtail, a small fly lands, sticks and is broken down where it lies until only a dry husk remains. This one let its glue do the slow work. The next kills with something you never see coming, light itself, and a single exit its prey can never find again. Number 3. The Cobra Lily, Darlingtonia californica. It rises from cold bogs like a hooded serpent frozen mid-strike, a translucent green and crimson head, a forked scarlet tongue hanging from its mouth. A nectar trail leads up to that tongue, but its role is debated. In one experiment, removing it did not change how much the plant caught. The real weapon, is light. The dome is riddled with translucent, chlorophyll-free patches, false windows that shine like open sky. The leading explanation is that a trapped insect flies toward them, batters against dead ends, and cannot find the one real exit behind the curl of the hood. As it tires, a waxy lining and downward hairs allow travel in only one direction, down. And this plant does not collect rainwater, it secretes and regulates its own pool. The insect drops in, and here is the horror. Armitage, in 2016, found the fluid is dense with bacteria that lower its surface tension to about 48 dyne per centimetre, well below water's 72, so prey breaks the surface and sinks. In his tests, raw pitcher fluid held every ant, the bacterial cultures held about 97% within 10 minutes, pure water held none. Digestion is largely microbial, with recent evidence of at least one of the plant's own enzymes. This one only had to wait. The next needs no maze and no trick of light, just a slick rim and a fall with no way back up. Number 4. The Trumpet Pitcher. Sarracenia, a tall funnel of living stained glass, laced with red and white veins, nectar beading along its rim. Nothing moves. Nothing looks like a mouth. Nectar draws insects to a lip that turns waxy and slick. Below, stiff downward hairs make every step forward easy and every step back impossible. Researchers have also identified conine, the alkaloid known from poison hemlock, in several Sarracenia species, generally in low or trace amounts. Its role is unresolved. Researchers have proposed it as either a lure or a paralytic, and it is not settled either way. Those beautiful red veins? A study of a pigment-free variety found capture-tracked leaf size, not color, so treat the veins as beauty, not a proven trap. The insect slips past the rim, tumbles into a pool laced with wetting agents that stop it floating free, and is slowly dissolved. And yet the pool is not death for everyone. The caterpillars of the pitcher moth Exera spend their whole larval lives inside these very tubes, grazing the inner wall and damaging the trap so it can never fill and drown them. Everything up to here has been patient, glue, false light, and pools that only had to wait. Number 5 is where these plants stop waiting. It moves and it counts before it strikes. Number 5. The Venus Flytrap. Dionea muscipula, the windowsill plant everyone underestimates, pink lobes rimmed with soft bristles like eyelashes. Inside stand trigger hairs. One touch does nothing. Bending a hair fires an action potential, the same kind of signal that travels a nerve, though the plant has no nerve and no brain. A second touch within roughly 20 to 30 seconds fires again, and only then does the trap usually spring. That two-touch rule is the leading explanation for how it ignores a landing raindrop. The snap is not muscle. The open lobes are held in a curved, pre-stressed state, storing elastic energy, and the trigger flips their geometry through a buckling instability that amplifies an underlying change in water pressure, releasing it in about 1/10 of a second. And it keeps tallying, on the order of 5 pulses before it commits to digestion. This is electrochemical bookkeeping. Not thought. The plant registers pulses. It does not decide. Sealed inside, the insect is digested over roughly 10 days. But the flytrap still needs to be touched twice. The next hunter drowns its prey before the walls ever close in. Number 6. The tropical pitcher plant. Nepenthes, a jewel goblet on a jungle vine, its glossy collar glistening like sugared glass, offering you a drink. Bone and Federle in 2004 showed that collar, the peristome, is a water-lubricated surface that becomes treacherously slippery chiefly when wet with nectar, rain, or humidity, so an insect's footpads aquaplane, like a tire on a wet road. When dry, it is far more secure. One misstep and prey slides into the pool. But the most unsettling twist, the giant montane species N. lowi and N. raja repurpose the trap into a toilet. Their pitchers offer exudate positioned so a tree shrew, or for N. raja, a summit rat, must straddle the mouth to feed and tends to defecate into the pool as it does. Clark and colleagues in 2009 estimated mature N. lowi draws 57 to 100% of its leaf nitrogen from those droppings. This is a mutualism. The mammal feeds and leaves unharmed. Nepenthes does not hunt mammals. Its everyday prey is insects, and vertebrate capture is rare and opportunistic. This one waits for prey to fall in. The next doesn't wait at all, it snaps shut around its prey underwater. Number 7. The Waterwheel. Aldrovanda vesiculosa, the flytrap's rootless aquatic sister, drifting just below the pond surface in whorls of tiny emerald clamshells. Each two-lobed trap, only a few millimeters long, is lined with roughly 40 trigger hairs. A water flea brushes them, an action potential races across the lobes, and they slam shut. But it does not close like the fly trap. Westermeyer and colleagues in 2018 showed it uses active hydraulics, a rapid loss of water pressure in the motor cells, plus the release of built-in press stress, not the fly trap's snap buckling click. The quick shutting phase is among the fastest movements in the plant kingdom, temperature and ecotype dependent, measured across roughly 10 to 100 milliseconds, the fastest around 10 to 20, about 10 times faster than its cousin. It seals aquatic microfauna, water fleas, copepods, mosquito larvae into a shrinking chamber and digests them alive for their nitrogen. It is IUCN endangered, down to about 50 wild sites, and yet the Waterwheel still has a jaw to close. The next hunter has no jaw at all, no snap, no speed. Its weapon is buried in the mud, and it never moves. Number 8. The Corkscrew Plant. Gentlest, above ground, almost nothing, a modest rosette small enough to hide in a teaspoon of moss. The weapon is buried. It has no roots. Instead, colorless corkscrew-shaped leaves spiral into the wet soil, each lined with rows of inward-pointing hairs aimed back toward a digestion chamber. For years, following work in the late 1990s, it was assumed the plant chemically lured prey. But the leading 2024 study on Gentlesea hispidula found no clear chemical lure. Microbes were neither attracted nor repelled. Instead, the passive geometry alone suffices, a one-way ratchet. A protozoan like paramecium swims in, and every random re-aim tends to nudge it deeper against the angled hairs. The plant does not pull, it does not pump. It builds a corridor that runs one way. Funneled to the vesicle, the prey is dissolved by glands. And Gentlest tuberosa holds the smallest genome ever measured in a flowering plant, roughly 61 million base pairs, yet still builds this trap. But even a corridor gives prey time to wander in. The next hunter gives no time at all. Number 9. The climax. The Bladderwort, Utricularia, the plant you'd never suspect and might never see. Rootless, often no thicker than a thread, it drifts in ponds and ditches on every continent but Antarctica, crowned with small snapdragon flowers. The killing apparatus is nearly invisible, hundreds of transparent bladders, most a millimeter or two wide, hanging like glass beads. Each is a spring loaded with emptiness. Glands actively pump water out until the internal pressure drops, roughly minus 16 kilopascals, varying by species, and the elastic walls cave inward, straining to relax. A prey animal brushes a trigger hair. Those hairs are not nerves, and the plant is not deciding. The leading explanation is that they act as mechanical levers, tipping the curved door past a point of elastic instability. The door buckles, snaps inside out, and the stored suction pulls in everything before it. According to Vincent and colleagues, in 2011, the whole suction is over in under a millisecond, roughly half a thousandth of a second, accelerating the inrushing water at up to about 600 times the force of gravity. Among the fastest movements known in the entire plant kingdom, and the fastest of any suction trap, the prey does not move toward the plant, the water moves inward and carries it. A water flea is simply inside before it can register that anything moved. And these traps also fire spontaneously, on their own rhythm, sweeping in prey too small to touch a hair. The prey stays microfauna, tiny crustaceans, larvae, protozoa, rotifers. That is the twist. The deadliest is not the biggest or the flashiest. After the giant Nepenthes goblet and a string of snapping jaws, the apex is a near-invisible thread strung with millimeter bladders, firing right now, on its own rhythm, in the water beside you. Look back at the climb. We began in beauty, a sundew wearing false dew, a buttery leaf, a serpent of light, stained glass in a bog. Then the plants began to move, a windowsill trap that tallies electrical pulses, a jungle goblet that runs a mammal latrine, an underwater snap, a buried one-way corridor. And it ended not in size, but in speed, half a millisecond, in a ditch you'd walk past without a glance. And not one of these plants set out to be a monster. They grew where the soil gives nothing, and every trap on this list is the same answer to the same hunger. If the ground will not feed you, the air will. If this rewired how you see the quiet green world, subscribe to the hostile host, because the next thing standing still beside you may not be as harmless as it looks. Host or hijacker. No one is safe.